Hey guys, so we are looking at section 2. We're going to talk about transport. So, how things move inside and outside of the cell. So, looking at the cell membrane. So, the cell membrane was that sac like structure that held all the organelles inside the cell. So, it's kind of like a wall or a barrier. So the cell membrane is a protective layer between the environment and then everything inside the cell, so all those organelles. And its job is to maintain balance. It's there to ensure that everything that's supposed to come into the cell, nutrients from the exterior, from the environment, do, and that everything that's inside the cell stays there and is where it's supposed to be. So there are two parts to the cell membrane. These terms will more or less haunt you in Bio 20 if that's something that you choose to take, in Bio 30. And so what they are is you have your extracellular membrane, so that's everything on the exterior, so that's space to the environment. And then you have your intracellular membrane, so that's going to be inside the cell. The membrane is made up of what we call a phospholipid bilayer. So it's made up of fat, that's the lipids. And so lipid is another term for fat. I don't know if you guys remember that from grade 9. And then phosphates. And so the phosphate and the fat actually make up the membrane. And there are two parts to the phospholipid bilayer as well, called the hydrophilic head. And so the hydrophilic head is actually it is attracted to water. And then the hydrophobic tail, which makes up the interior of the membrane, so this wall, is hydrophobic. So it'll actually repel water. Okay, so a phobia is that you're afraid of something, so it's a hydrophobic tail. So this membrane is a lot more complex than just what we see or just drawing a line to shape out the exterior of a cell. It is made up of special cells, and it does control the input and the movement of things inside and outside of the cell. So a term that you might have read about in your textbook is semi-permeable. So if something is permeable, it means that material can flow through it. Sand, for example, is a permeable material, so water can easily flow through the grains of sand. So semi means partial. Okay, so semi-permeable membrane, based on the size of these molecules. So here are some red blood cells in the picture and some other maybe inorganic substances. So it's permeable, these holes within the membrane dictate what goes inside and outside the cell. And so based on size, primarily, certain things are allowed in and certain things just can't enter. So that's defining a semi-permeable membrane. So based on the size of the molecule, they can either enter or exit that cell membrane. Selectively permeable also looks at essential nutrients and molecules and how they can pass through. And so depending on what the molecule is, if it's sodium or potassium, based on the importance, based on the concentration. So if there's a lot already in the cell or if there isn't, then the cell can become selectively permeable. So it can almost more or less choose what enters and exits the cell based on need. And mostly concentration is how it works, but it comes across like need. So a little bit of a review from the particle model of matter. When we look at solid material, when we look at solid matter, the particles are very closely knit together and they actually vibrate against one another. And so the idea that particles are always moving. So at a solid state, particles are closely knit, and as we change state, as we go from a solid to a liquid to a gas, the particles actually start to move faster and they start to vibrate or kind of shift and shuffle and exchange places. And in a gas, they'll actually take up the space of the container and they're just bouncing around and moving as fast as they can. And so this concept of solid, liquid, and gas, the particle model of matter, is important when we look at how particles move. And so, and concentration and what's going inside a cell versus outside the cell. So there's a video I'd like you guys to watch about diffusion. And so diffusion is something you guys would have learned about way back in grade 8. And so diffusion is this passive movement of particles. So no energy is required in order for these particles to move. And so when we look at diffusion, an example is if you sprayed some perfume, or if your mom sprayed some perfume in the room next to you, eventually you would smell it. And that's because particles are moving from a high concentration down here to a low concentration. So they're always moving to where there's extra space, to wherever they're needed, to that lower concentration. So particles here on the left-hand side would move to the right because they're highly concentrated to the left, and they're going to move from high to low. And they're just going to do that passively. No energy is required. We can increase the rate of diffusion. So another example is if you were to put food coloring in a glass of water, you would notice that without even touching the glass, eventually the food coloring would disperse, and it would evenly disperse within the water. And that is diffusion. No energy was required for that to happen. The food coloring is concentrated, those particles, and so they're gradually going to disperse, move from high concentration to low, and create that equilibrium, create that balance. So, but we can increase diffusion, and some common methods are by heating up a substance. So when you add energy, when you heat something up, you're like going from a solid liquid to a gas, you're adding energy. And so by adding energy, by adding heat, thermal energy, you're increasing the movement of those particles. And so they're going to move really, really fast, and so they'll quickly disperse. And so that's one way you can speed up the rate of diffusion. Another one is stirring a substance. So when you add sugar to your tea, you most often stir your tea because that allows for that sugar to quickly dissolve within your substance, within your tea or hot water. But if you were to leave it, it would gradually disperse and diffuse on its own as well. So it's really important to know some ways, some processes that we can increase the rate of diffusion, heating up a substance, adding energy, and stirring, and actually mixing or moving that substance would help. So diffusion, moving through this selectively permeable membrane, so allowing for certain substances to move from one side to the other across this phospholipid bilayer. It's important to know that particles are always, always, always going to move from high to low concentration in the processes of diffusion and osmosis. So think of yourself in a really crowded, in a mosh pit, or in a room, or you're in a really long line, and then a shorter line opens up, or there's just some space in the crowd. So you naturally would probably go to the shorter line, or you would go to that open space in the crowd to alleviate yourself, right, from all that tension. And so particles are going to do that as well. They're going to move from high to low concentration. Concentration gradient is an important concept for Bio 20. And so what it is, it determines the direction of all these things, if things are going to be moving. And so based on, we're trying to create balance. Okay, so all cells want balance. They don't want too much of something, or they don't want too little of something. And so this concentration gradient, what it refers to is the direction that molecules or particles are going to move. So it's going to dictate that it's more concentrated on one side and that there's a lower concentration on the other. And so whatever that barrier is, typically a cell membrane, we call that barrier the concentration gradient. So it's there, and it's dictating, or it's defining two sets of concentrations, one that's high and one that's low. Okay? So it's not actually a structure, the concentration gradient. For example, this is our semi-permeable membrane, or a selectively permeable membrane, but it also outlines kind of like this invisible term or concept known as the concentration gradient. Because there is a barrier, you've created a gradient, a concentration gradient. So one side is more concentrated than the other. And the goal for cells is to balance this, okay, to find what we call equilibrium. Okay? Again, a selectively permeable membrane is looking at the movement of these particles. And so based on concentration gradient, based on what a cell might need, if it's needing salts or potassium or water, those particles are going to move across that concentration gradient. Okay. So some characteristics for your concentration gradient, it must involve different concentrations in order for us to create this invisible boundary. It drives diffusion and osmosis. And so what that means is that because diffusion is passive and so is osmosis, which I'll talk about, these particles and substances are always going to move across a concentration gradient. They're always going to move from high to low. And so those concentrations, that difference is important because it dictates the movement of those particles. Different molecules or ions move along their own concentration gradient independently. So again, like if we're looking at salt, sodium, or just sodium is going to have its own concentration gradient. It's going to look at itself and say, is there more salt on one side of the cell versus the other? It's not going to look at all the different ions or particles in the area. It's just going to look at salt. So do you need more salt or do you not need more salt? Do you need more water? Do you not need more water? So molecules work independently on their own, based on their own concentration gradient, not as a collective of stuff. In cells, different concentrations may be separated by the membrane. So there are some membranes that are selectively permeable, and again, they'll allow for only certain substances. And so maybe they don't allow for the movement of salt within that cell, within that structure. And it's always going to involve molecules or ions of a single type. So this process of osmosis, you learned about it for the first time in grade 8. It is also a passive, no energy is required process. And what we're looking at is the concentration of salt and water. And so it's the movement of water across the membrane. And water is always, again, going to move from a high concentration gradient to a low water concentration gradient, or it'll move from a not so salty place to a salty place. So think of it, or the way I think of it, is water moves to where it's needed. Okay, and so if I gave you a salt tablet, or if I just asked you to eat a bunch of crackers that were super, super salty, or French fries, you would eventually feel thirsty. You need water. And so when there is a lot of salt in an area, there also needs to be a lot of water in order to dilute that solution. And so water is then going to move to that salty area. So if we look at the image here, you'll notice that the blue are all the water particles, and then the green are the salt. And so what's going to happen is water is actually going to move from the left to the right. It's going to move to the saltier concentration in order to help dilute and kind of balance that solution. And that's osmosis, the movement of water. Okay, so there is a video that I would like you to watch related to osmosis. And so again, it's just moving from that high water concentration to low water concentration, or also known as low solute. So that would be the salt. To a high solute concentration. So water is going to go where it's super salty. Tonicity refers to some terms looking at cells and the movement of water and that equilibrium. So if something is hypertonic, if a solution is hypertonic, what that means is that there's a lot of salt in that solution, lots of salt. And what's going to happen to cells is all the water from the cells is actually going to leave, and it's going to go into that salty solution. And so cells can rupture, break, and die because of this. So this is why, like, you'll never put freshwater fish in salt water because they'll die. And this is how they die. What happens is the water from inside their body goes to the saltier exterior and they become dehydrated and die. A hypotonic solution is when a cell is actually more salty than the exterior. And so this is also why you would never put a saltwater fish in fresh water, because the interior of the saltwater fish is salty. It has a certain concentration. And what's going to happen is all the water from the fresh water is going to then go into the fish and it'll explode and die. So having that balance is really important, and that's why there are designated fish for saltwater and freshwater. Same thing for our own cells. You might notice that if you do go out into the salt water, into the ocean, or if you were to put your hand in a lot of salt, eventually it would dry up. And that's because all the water and moisture in our body from our cells will go to that salty exterior. And so that would be hypertonic. So unfortunately, these are animations, but they do show you that in a hypotonic solution, where the cell is saltier than the exterior, water would go in and then it would become really big and possibly rupture. And another term is called lysing. In a hypertonic solution, it's super salty around the cell, and so water is going to leave and it's going to shrivel. In an isotonic solution, this is where we have balance. And so that's the goal, to have balance and to be in an isotonic solution. So the exterior is close or similar to our interior. Okay, so these are, again, other examples or images. Isotonic, you see that there's balance. Hypotonic, the salty cell, water's going in and it'll rupture and get really big. And then hypertonic, the red structures are the salt, and so water is leaving. Okay. Another term that you need to know, and so there is a video related to tonicity that I would like you guys to watch, but another term is active transport. So active transport requires special molecules or enzymes to assist in the movement of material. So there are special enzymes that are going to be within this phospholipid bilayer, within the membrane, and they assist and help with the movement of stuff. But the catch is that these enzymes require energy. And so energy is actually needed in order to get stuff moving inside and outside of the cell. And so that is a big difference between diffusion and osmosis. Diffusion and osmosis are passive. They look at that concentration gradient, whereas active transport requires energy, and we have these special enzymes that allow for the movement of material. Okay. Facilitated diffusion and channel proteins and carrier proteins, they're all really similar. But so what's happening here is we have proteins, again, like enzymes, that are within the cell membrane, and they help with the movement of all these particles. Okay. Two more terms. Another one is endocytosis. So endocytosis is the process where we talked about vesicles, and you had drawn vesicles in your plant animal structures. They're like little packages with material going inside and outside the cell. So endocytosis is a process when the cell takes in material, so endo coming into the cell, and we have these vesicles that are then going to go to the Golgi bodies, and they're going to kind of relay what they have. So endocytosis is coming from outside the cell to inside the cell through vesicles. And then endocytosis, and this is an animation that would typically show you endocytosis coming inside the cell, and then exocytosis exiting, where these vesicles come to the surface of the cell membrane and then they release their material. So here's exocytosis, this cell with maybe these different proteins or enzymes or chemicals or hormones, it's going to come to the surface of the cell membrane and then it'll release those particles out into the environment or out into a different cell. Okay, and so that's the section for this week, or my little lesson. I do have some readings for you guys in the textbook and some check and reflect that I would like you to complete and some videos that I'd like you to watch. So please let me know if you guys have any questions and have a good one. Bye.